Plants living in variable soils face a real strategic dilemma, and this slide shows it clearly. Under rich conditions, roots spread shallow and wide to scavenge nutrients. But when drought or salinity hit, the hormone ABA triggers a shift toward narrower, deeper roots to access water and reduce loss. The image contrasts those two architectures and highlights the trade-off between nutrient foraging and survival. This page maps how ABA is made and moved around the plant, showing leaf synthesis, rapid phloem transport, and local root production via NCED3 when stress appears. The diagram also points to specific transport gates like NRT 1.2 and ABCG 25 that control systemic ABA flow, so the whole plant can rapidly rewire growth and water balance in response to signals. Think of ABA signaling as a molecular gearbox. In the no-stress frame, PP2C phosphatases lock the engine and SNRK2 stays inactive, keeping stress genes off. When ABA accumulates, it fits into PYL receptors like a key, sequestering PP2Cs and freeing SNRK2 to phosphorylate downstream factors, flipping the switch to activate stress-responsive transcription and physiological defenses. This slide focuses on spatial precision, showing how ABA acts differently across root tissues to fine-tune growth. In the elongation zone, moderate ABA tempers ethylene to preserve primary root elongation. The endodermis ramps up suberin deposition to prevent radial water loss, and the quiescent center limits stem cell divisions to protect the niche under severe stress. Each tissue contribution is targeted and complementary. Here we see how ABA negotiates with other hormones to shape root architecture. It synergizes with mechanisms that sustain primary root growth while antagonizing auxin-driven lateral branching, ethylene synthesis, and cytokinin signals that promote division. That crosstalk allows the plant to prioritize deep-water foraging and stress tolerance over energy-intensive lateral proliferation when resources are scarce. The summary table on this slide contrasts well-watered and drought-adapted root architectures, showing the functional outcomes of ABA action: sustained downward primary root growth, suppressed lateral root density in dry topsoil, increased root hair elongation for microabsorption, and enhanced suberin deposition to reduce water loss, all combining to optimize resource acquisition under stress.